Thank you very much, Marcus, and thank you to the Emanuela and the other organizers for inviting me to speak at today's meeting and for this opportunity to visit this beautiful city of Trieste for the first time. So, in the next 30 minutes, I'm going to provide you with an overview of the molecular neuropathology of frontotemporal dementia, focusing on the findings from human postmortem studies. I will try to provide some updates on recent developments, and then I'm going to finish by summarizing our recent findings related to TIA1 as a possible new FTD and ALS causing gene. So as you're aware, the neuropathology underlying clinical frontotemporal dementia is heterogeneous. Degeneration of the frontal and temporal lobes is a relatively consistent feature, and the term frontotemporal lobar degeneration, or FTLD, is often used as an umbrella term for those pathologies that commonly present as clinical FTD. As with many other neurodegenerative diseases, the pathology of most cases of FTD is characterized by the abnormal intracellular accumulation of some disease-specific protein in the form of inclusion bodies. And a few years ago, we introduced this new classification where cases are initially placed into broad categories based on the molecular defect that is felt to be most consistent, or most characteristic, I should say. And then more traditional disease names are still used to define more specific pathological patterns of pathology. And so in the current version of this classification, as you can see, there are three major molecular categories and a little bit of a garbage bin over here on the right, with the vast majority of cases of FTD having either tau or TDP43 pathology. So starting first with the tauopathy group, these conditions, there is overlap in the pathology of these conditions. However, each can be distinguished based on the specific morphological subtype of the tau immunoreactive inclusions that form. And so with PIC bodies are the defining feature of classical Pick's disease, whereas corticobasal degeneration, tau accumulates in the distal processes of astrocytes, producing these astrocytic plaque-like structures, and in PSP you get these tufted astrocytes. In addition, there is variation in the biochemical species that forms these tau immunoreactive inclusions in the different conditions, so that PIC bodies are composed predominantly of 3-repeat tau isoforms, whereas most of these other types of inclusions are composed predominantly of 4R tau. The neuropathology associated with MAP tau mutations varies depending on the specific mutation. So those mutations that affect the alternate splicing of exon 10 tend to result in an overproduction of 4-repeat tau, and they tend to get pathology that is similar to the sporadic 4-repeat tauopathies, PSP and CBD, whereas mutations in other exons tend to result in inclusions that contain both 3R and 4R tau that may more closely resemble the neurofibrillary tangles of Alzheimer's disease. Now, the most recently characterized of these tauopathy conditions is this globular glial tauopathy, and so I'll just spend a few minutes highlighting it in case it's one that you're not familiar with. So this term globular glial tauopathy has recently been recommended for a group of uncommon sporadic 4R tauopathies that are characterized by these tau immunoreactive globular cytoplasmic inclusions within oligodendrocytes and astrocytes. This pathology predominates within the frontal and temporal lobe white matter, with lesser involvement of the frontal and temporal cortex and basal ganglia. The clinical phenotype ranges from pure FTD, which is usually the behavioral variant, to pure upper motor neuron degeneration or primary lateral sclerosis, or these two phenotypes can be combined. Now, this is not a new entity. Cases of this condition have been published previously under a variety of terminologies. Sporadic multiple system tauopathy with dementia, for instance, is one of those. And in addition, these globular glial inclusions can occasionally be seen as part of the pathology associated with some MAP tau mutations. Another recent development has been the growing appreciation of tau immunoreactive astrocytic inclusions as an incidental finding in aging. And the term aging-related tau astrogliopathy, or RTAG, has been coined to describe primarily two different types of inclusions shown here at the bottom. These thorn-shaped astrocytes in which there is dense perinuclear tau immunoreactivity that extends into the proximal processes, giving these sharp protrusions that are sort of like rose thorns, and these granular or fuzzy astrocytes in which more granular tau immunoreactivity extends into the distal processes of astrocytes. The thorn-shaped astrocytes tend to predominate at the glial limitans, in the subpial, subependymal, and perivascular regions, as well as in the cerebral white matter, whereas the fuzzy astrocytes tend to predominate in the gray matter, either in isolation or in clusters. Now, it's usually possible to distinguish these age-related astrocytic inclusions from, on morphological basis from those that characterize the primary tauopathies. However, RTEG can coexist with these and other age-related neurodegenerative diseases and can potentially cause diagnostic confusion. And recently, Gavor Kovacs has established an international consensus group of neuropathologists to more formally study RTEG to try to determine its clinical and biological significance. Now, in most FTD series, the largest group are those characterized by pathology composed of TDP43. And these cases can be subclassified or subtyped based on the relative proportion of different types of tau immunoreactive neuronal inclusions and their laminar distribution within the neocortex. And each of these different subtypes tends to have relatively specific clinical and genetic correlations. However, what's often forgotten is that the original description of these subtypes was made prior to the discovery of the pathological protein in the tau-negative FTLD cases and was based on the findings with ubiquitin immunohistochemistry. Once it was recognized that most cases of FTLDU are TDP43 positive, the original ubiquitin-based criteria were simply translated for use with TDP43 immunohistochemistry without any formal validation. And there are a number of potential problems with this. First of all, it turns out that not all cases of tau-negative FTLDU have TDP pathology. Some we now know have FET protein pathology, which I'll describe in a moment. TDP immunohistochemistry is very sensitive and demonstrates some additional pathology that is not well recognized with ubiquitin and would not have been included in that original assessment. And then finally, there are some cases of FTLD TDP that have additional ubiquitin-positive pathology that is not TDP immunoreactive. And this includes the dipeptide repeat pathology in cases with the C9orf72 mutation. And so these factors could lead to discordance between the findings seen with ubiquitin immunohistochemistry versus TDP immunohistochemistry, and potentially lead to inconsistencies or confusion in subtyping cases based on TDP. And so to address this, Manuela Neumann and I recently performed an unbiased semi-quantitative assessment of the TDP immunoreactive pathology in cases following the original FTLDU criteria. So what we did is took our 78 cases and originally subtyped them as type A, B, or C based on the findings with ubiquitin immunohistochemistry using the original criteria. We then performed a blinded semi-quantitative assessment of the patterns of TDP pathology using both phospho-TDP antibodies and phosphorylation-independent TDP antibodies, which tended to show relatively similar findings. And what we found was that the type A and type C cases tended to show very similar findings with both TDP immunohistochemistry and ubiquitin. So the type A cases are characterized by dense neuronal cytoplasmic inclusions and short, thick neurites, which are concentrated in lamina 2, as well as at least some of these small lens-shaped neuronal intranuclear inclusions, whereas the type C cases are characterized by a predominance of neuritic pathology, which often includes many of these long, tortuous neurites, and which may either be superficial or concentrated in deeper cortical lamina as well. However, with the type B cases, there were some potentially important differences. So with ubiquitin immunohistochemistry, type B cases are characterized by rather modest numbers of neuronal cytoplasmic inclusions throughout all cortical laminae, with a relative paucity of neuritic pathology. With TDP immunohistochemistry, these cases showed more cytoplasmic inclusions and demonstrated that the majority of these actually have this granular appearance that wasn't really appreciated with ubiquitin immunohistochemistry. In addition, many of these type B cases show abundant delicate wispy threads and dot-like profiles that are TDP immunoreactive and may be either pan-cortical or in a laminar distribution. And this is important because sometimes this thread pathology is concentrated in layer 2, where it may create a superficial laminar pattern that is reminiscent of the type A cases. And so based on these findings, we've proposed some rather minor revisions to the pathological criteria for FTLDT subtyping. The first is to appreciate the differences in the quantitative differences in the nuclear cytoplasmic inclusions, with compact inclusions being much more characteristic of the type A cases, and the type B cases being characterized by predominance of more granular cytoplasmic inclusions. And then secondarily, that the presence of this thread and dot pathology, although not absolutely consistent, when it is present is highly suggestive of type B pathology. So the next stage is to validate this in a multi-centered analysis and see whether these minor revisions improve the consistency in subtyping amongst observers. Now, another interesting finding that came out of this study was that a significant number of our cases actually showed the positive pathological features of both type A and type B cases. So these cases have, again, the dense cytoplasmic inclusions and the short, thick neurites, as well as some of these lentiform internuclear inclusions that are typical of type A cases. But they also show significant numbers of neuronal cytoplasmic inclusions in deeper cortical lamina, which often have this granular morphology. And some of these cases even showed that wispy thread and dot-like pathology characteristic of the type B cases. Interestingly, the majority of these cases turned out to harbor the C9orf72 mutation and presented with a combination of FTD and ALS. And the remaining 3 cases were all sporadic cases of FTD plus ALS. So the appropriate classification of these cases under the current system is perhaps problematic, but the relatively consistent genetic and clinical correlation suggests that these may represent a unique subset and may also explain why the pathology associated with the C9orf72 mutation has inconsistently been reported as either type A or type B, depending on how the observer rates the various positive and negative features in determining the subtype. Now, before leaving the C9orf72 mutation cases, I just want to say a few words about the dipeptide repeat pathology. So when this mutation was originally discovered, we proposed 2 different potential pathomechanisms: partial reduction of protein function due to suppressed translation of the mutant allele, toxic gain of function due to the formation of RNA foci composed of the expanded transcript. However, following the subsequent recognition that the expanded repeat could undergo RAN translation and develop 5 different dipeptide repeat peptides, an enormous interest has developed in the potential pathogenic role of dipeptide neurotoxicity in this process. And a number of speakers later in the conference will be discussing each of these potential pathomechanisms. But I want to just summarize 2 studies that Manuela and I published focusing on the findings in human cases. So in this study, we looked at a series of C9orf72 mutation carriers that spanned the clinical spectrum from clinically pure ALS to clinically pure FTD. We found that the anatomical pattern of neurodegeneration closely correlated with the phenotype, as you would expect. So cases of ALS have predominant degeneration in the spinal cord and the lower brainstem, whereas cases of pure FTD have predominant degeneration of the premotor frontal cortex. The distribution of TDP pathology closely mirrored that of degeneration and correlated with the phenotype. However, the anatomic distribution of the dipeptide pathology showed no correlation with either the phenotype or degeneration. It was consistently high in areas that rarely showed any degeneration, such as the occipital cortex and the cerebellum, and was consistently negligible in the spinal cord, even in cases with clinical ALS. And this lack of clinical pathological correlation was true when we looked at all 5 of the different dipeptide repeat pathologies. And so our conclusion from this was that the neurodegeneration associated with the C9orf72 mutation is more closely associated with TDP mismetabolism. And although the presence of the dipeptide repeat pathology is an absolutely consistent and specific feature that identifies the presence of the mutation, its pathogenic role is still somewhat controversial. Now, having said that, I'll recognize that these findings may simply represent the limitations of studying end-stage pathology using tools as blunt as light microscopy and immunohistochemistry. Nonetheless, I think that any models proposed based on experimental studies need to incorporate these findings, both the absence of visible dipeptide repeat inclusions and the presence of TDP-positive aggregates within the degenerating cell populations. The third major molecular group are those characterized by inclusions that are immunoreactive for FUS and the other FET proteins, Ewing sarcoma and TAF15. And these include a group of related but distinct clinical pathological conditions that include atypical FTLDU, which tends to present as early onset behavioral variant FTD in the absence of motor features, and neuronal intermediate filament inclusion disease and basophilic inclusion body disease, both of which may present with various combinations of FTD, pyramidal, and extrapyramidal motor dysfunction. Importantly, unlike cases of ALS caused by FUS mutations, in which the pathology is only immunoreactive for FUS and not the other FET proteins, these cases of FTLD FET are always sporadic and not associated with any mutation. Now, the only point I want to make regarding this group relates to the phenotype associated with the atypical FTLDU pathology. These cases were originally identified based on the presence of this unique molecular pathology consistently associated with a rather unusual clinical phenotype. Very early onset, severe psychobehavioral abnormalities, often antisocial, violent, even criminal behavior, that was rapidly progressive. All of our patients were permanently institutionalized within 2 years of disease onset. And at postmortem, the brains tended to show severe atrophy, usually weighing less than 1,000 grams, and the FUS and other FET pathology was anatomically widespread throughout the central nervous system. However, here is a case that I recently reviewed. This gentleman was perfectly well until age 68, at which time he developed slowly progressive, mild behavioral abnormalities that included apathy, restlessness, and obsessions. When he died 5 years later, his brain was only mildly atrophic, and the FET protein pathology was very mild and restricted basically to the neocortex and the hippocampus. And so, although this may represent an atypical case of atypical FTLDU, it nonetheless illustrates that the phenotype may be more variable, and it may not always be possible to predict the presence of the underlying FET pathology during life. Okay, I'm going to finish with a brief summary of our recent evidence supporting mutations in the TIA1 gene as a cause of FTD and ALS. And this started with a study of one of our UBC families known as family U2. This is a family of European ancestry with autosomal dominant inheritance of ALS plus or minus dementia. Pathology on 2 deceased affected individuals showed TDP pathology in the brain and spinal cord, which I'll show you in just a moment. Our initial genetic analysis excluded mutations in C9orf72 and other known ALS and FTD genes. And so we performed whole exome sequencing on these 2 affected second-degree relatives and identified candidate causal mutations by filtering the identified variants using fairly standard criteria. This resulted in 15 genes with variants, of which 5 had CAD scores greater than 20, indicating that they are likely to be highly deleterious or damaging. And our attention was immediately drawn to one of the most highly ranked variants, which was this P362L mutation within the TIA1 gene. T-cell restricted intracellular antigen 1 is an RNA binding protein similar to other ALS and FTD associated proteins, such as, there we go, got it, such as TDP43, FUS, HNRMPA1. It contains multiple RNA recognition motifs and the C-terminal prion-like low complexity domain, which is required for the protein's self-assembly and the formation of membranous organelles via the process of liquid-liquid phase separation, which is a common feature of our RNA binding proteins that we'll discuss in just a moment. More specifically, TIA1 is the main molecular driver behind the process of stress granule formation. And dysfunctional stress granules have been implicated in neurodegeneration in general and ALS in particular. A study by Aaron Gittler in 2011 actually identified TIA1 as a candidate ALS gene based on structural and functional homology with other RNA binding proteins. And the P362L mutation affects a highly conserved residue within the low complexity domain that is predicted to be highly deleterious or damaging. In addition, a different mutation within the low complexity domain of TIA1 was previously reported to cause a different disease within Swedish and Finnish patients known as Wielander's distal myopathy. So this is a disease of muscle characterized by rimmed vacuoles which contain aggregates of TDP43 and P62. And these are very similar to the inclusion body myopathy which you may encounter in patients with mutations in either VCP or sequestosome, which may also cause ALS and FTD. And so for these reasons, we prioritize TIA1 as a candidate ALS FTD gene. We performed a genetic association study in which we sequenced the exons encoding the low complexity domain. We identified 6 additional variants in just over 1,000 samples from unrelated ALS patients, none in over 3,000 controls, giving a p-value of 8.7 times 10 to the minus 6. These variants were all either absent or extremely rare from the exact database, and all had a CAD score greater than 20, again suggesting that they are highly deleterious. And so this represents a mutation frequency of 2.2% of our familial ALS cases and 0.4% of our sporadic ALS cases, similar frequencies to what you see with sequestosome and VCP, for instance. Here's a summary of our 9 mutation carriers, 3 from our UBC family, 6 unrelated relatives. Disease tended to present in mid to late adult life and presented as either focal weakness or aphasia. All of the patients went on to develop ALS, and half developed features of FTD. There was one outlier here. Our UBC U214 patient had a rather unusually young age of onset, and interestingly, she had childhood dyslexia that she probably inherited from her father, who is also dyslexic, and so she may actually have a double genetic hit. A family history was present in 3 of 7 probands, and interestingly, all of these individuals were female, although we did have some affected male family members within UBC U2. In addition to our 2 UBC cases, postmortem pathology was also available from 3 additional patients. And the pathology on these 5 different individuals carrying 4 different TIA1 mutations was highly consistent, supporting a common underlying genetic abnormality. Degeneration of the neocortex was accompanied by TDP43 positive inclusions that were granular in nature with a relative absence of neurites, so this is type B. There was more widespread subcortical involvement affecting the basal ganglia and the substantia nigra. And in the spinal cord and lower brainstem, we saw the typical features of ALS, with degeneration of corticospinal tracts, loss of lower motor neurons, and various types of TDP43 immunoreactive inclusions. A somewhat unusual and consistent feature in these cases, which may represent a pathological marker for this mutation, was the presence of these large, round, hyaline Lewy body-like inclusions in some of the remaining lower motor neurons. We also performed immunohistochemistry using a number of different commercial antibodies that recognized different TIA1 epitopes and found no abnormal pattern of staining. We also performed double-label immunofluorescence with an antibody against phospho-TDP labeled in green and various TIA1 antibodies labeled in red, and again saw no colocalization of TIA1 to the cellular inclusions. And consistent with this, protein extracted from the brains of the TIA1 mutation carriers did not show any enrichment of TIA1 in the insoluble fraction. So to the best of our ability, we do not see TIA1 as a component of the cellular inclusions at the end stage of disease. So the next thing we wanted to do is look at the functional effects of these disease-associated variants, specifically on how they might affect stress granule dynamics. The stress granules are a member of this larger family of membranous organelles known as ribonuclear protein bodies. These represent complexes formed between RNA and RNA binding proteins. They exist as non-membrane bound compartments in the nucleus or the cytoplasm that provide spatial separation and a unique chemical environment that promote various types of nucleic acid remodeling. And they're important for the storage, processing, transport, and degradation of RNA. These structures form in response to various physiological stresses, using the process of liquid-liquid phase separation, which is dependent upon the biochemical properties of the resident RNA binding protein, which self-assembles in a concentration-dependent fashion to form liquid droplets that are held in a distinct phase from the surrounding medium via multivalent forces. And you can picture these as being like small droplets of oil suspended in water. And these structures form and disassemble rapidly in response to changing environmental cues. So the first thing we wanted to look at was the effect of the mutations on the biophysical properties of TIA1. We found that full-length wild-type TIA1 spontaneously phase separates at room temperature to form these liquid droplets, and that the purified mutant TIA1 also undergoes spontaneous concentration and temperature-dependent phase separation to form droplets that are morphologically indistinguishable from those from the wild-type TIA1. However, the difference was that the mutant proteins caused a significant leftward shift in this coexistence line to a lower concentration, meaning that they promote liquid-liquid phase separation, probably due to increased intramolecular forces. We also looked at how these mutations affected the mobility of the protein in this complex multi-phase fluid environment using a process known as fluorescence recovery after photobleaching. So solutions of wild-type and mutant TIA1 were cooled to the point where they underwent phase separation and formed droplets in which the fluorescence-tagged TIA1 was concentrated. Individual droplets were then laser photobleached, and you can see them surrounded by a yellow line here. These were photo-laser bleached, and the time it took for those droplets to spontaneously recover their fluorescence was measured as an indicator of how readily the protein is able to move back and forth between the dense droplet phase and the more dispersed phase. And what you can see here is that each of the mutations caused a significant change in the dynamic exchange of the protein between the two phases, as evidenced by this prolonged recovery time. Again, indicating that the mutants have changed the material properties of the droplets, probably because of increased intermolecular interactions, and this has impeded the protein mobility. Next, we wanted to see how these biochemical changes actually affected stress granule dynamics within living cells. And to do this, we generated a live cell assay which allowed for the real-time video monitoring of the kinetics of stress granule formation and disassembly following a heat shock. What we found is no difference in the formation of stress granules. However, each of the disease-associated variants resulted in stress granules with a prolonged disassembly time. So they failed to disassemble, with many stress granules persisting for up to 3 hours after the original stress. And then finally, we wanted to see how these purely dynamic persistent stress granules could eventually lead to the formation of TDP-43 pathology. And it's known that TDP-43 is one of the proteins that is consistently recruited into stress granules, and we confirmed that cells expressing both wild-type and mutant TIA1, that TDP is colocalized to stress granules following stress. We then performed a series of biochemical studies in which we found that in resting cells that had not been stressed, that TDP-43 was primarily recovered from the soluble phase, shown here on the left, with very little in the insoluble phase. However, cells that were stressed for 30 minutes, the TDP recovered from those showed a significant shift from the soluble phase to the insoluble phase. However, the shift in solubility was reversible. So if you allowed the cells 3 hours to recover, then the TDP-43 had largely returned to the soluble phase. Importantly, however, this reversibility was dependent on the duration of the original stress, so that if we doubled the stress to 1 hour, then TDP became permanently insoluble. Even after 3 hours of recovery, TDP remained in the insoluble fraction. And so what this implies is that these prolonged stress granules that stick around may provide an environment in which TDP can start to convert into a stable insoluble form. So to summarize this study, we have genetic evidence implicating mutations in the low complexity domain of TIA1 as a genetic cause of ALS and FTD. This is supported by the presence of a consistent and somewhat unusual neuropathological signature. Our biochemical studies show that the mutations result in increased intermolecular interactions which promote liquid-liquid phase separation and impede protein mobility. which in turn results in altered stress granule dynamics with stress granules that fail to disassemble as rapidly as they should, and that these persistent stress granules then provide an environment in which TDP can begin to aggregate and form insoluble inclusions. This highlights the central role of RNA metabolism in ALS and FTD and places stress granule dynamics at the center of ALS and FTD pathogenesis. And finally, it adds TIA1 to the growing list of genes responsible for a group of inherited pleiotropic neurodegenerative diseases known as multiple system proteinopathies, which may variably affect the nervous system, muscle, and bone. And so in summary, here again is the molecular classification for FTLD pathology with the major genetic and clinical correlations. As Peter pointed out, each of the genes is consistently associated with a particular molecular pathology, but unfortunately for the sporadic cases, there is much less consistent clinical pathological correlation. There are some relatively consistent associations. So for instance, cases of semantic dementia are almost always found to have FTLD TDP type C. Cases of FTD combined with ALS almost always have underlying TDP pathology. But for the most common presentations, which are sporadic behavioral variant and sporadic progressive non-fluent aphasia, it's impossible to predict the underlying molecular pathology in vivo. And for this reason, we really need good biomarkers if we're going to study and perform clinical trials on these patients. And so I'll finish there, and sorry I ran a few minutes over time.